All right, folks, welcome back. Today we're going to look at what's called protein synthesis, and that is how does DNA code for the proteins that function in our body. They do a whole lot of different things. They serve for structural purposes. They serve as enzymes that make chemical reactions take place. They do a lot of different things in our body, so it's really important for us to understand how do those proteins work, because those proteins essentially make up who we are as individuals. All right? So that's kind of what we're going to be looking at today, is a little bit about protein synthesis. How does it work? So today we're going to be analyzing what's called the central dogma of biology, which explains how DNA functions inside the body. You're also going to be able to describe protein synthesis in two steps, one known as transcription and the other known as translation. One comes before the other. Way you remember that is that they're alphabetical. Transcription comes before translation. All right, so let's get started. The central dogma of biology simply describes the process of how proteins are made. We start with DNA. The DNA is transcribed to make RNA. RNA is then translated to make proteins. So this step-by-step process is called the central dogma. We start with DNA, make RNA, and then we make protein. DNA, transcription to RNA, RNA translation to protein. So that's what we're going to look at today, is the central dogma of biology, one of the most important and key concepts in learning biology. In order to understand this, we need to take a look at RNA and understand why RNA is important, not only how it's similar to DNA, but also how it is different as well. There are a few key differences, though. RNA is single-stranded, where DNA is double-stranded. So there's one key difference, that DNA comes with two strands that are hydrogen-bound together through the nitrogen bases. RNA is simply single-stranded. Typically, it's not found as double-stranded. RNA has no thymine, so instead of ATCG, RNA pairings are going to be AUCG. A little bit different. We have uracil instead of thymine now. Additionally, RNA contains ribose sugar, whereas DNA contains deoxyribose sugar. So there is a big difference here between DNA and RNA. The RNA is single-stranded, has uracil, and contains ribose. There are several different types of RNA that we want to look at. Two of them are known as mRNA and tRNA. mRNA carries the DNA code to the ribosome. So mRNA is what's going to be made inside of the nucleus. It's going to take that code, those A's, T's, C's, and G's code, from the DNA and take it out to the ribosome that's in the cytoplasm of the cell. tRNA is specifically found on or near the ribosome, and that carries the amino acid. Remember that amino acids are building blocks of proteins, so tRNA is going to match up with mRNA and leave behind amino acids. We'll discuss this process in a little bit more detail later on, but remember that there are two types of RNA, mRNA and tRNA. There are two steps to protein synthesis, and two processes involved to make that protein. The first is called transcription, where we take our DNA and convert it into mRNA. And the second is known as translation, where we're going to take that mRNA, read it, and then make proteins out of it. So there are two key different processes here that are involved in protein synthesis, each one extremely important in order to make a fully functioning protein from DNA. The process of transcription is very similar to replication. The only difference is that instead of making a complementary strand of DNA, we're going to be making a strand of mRNA. So the first step is that DNA is unzipped using DNA helicase, and then instead of DNA polymerase, like we would find in replication, we're going to use RNA polymerase to make mRNA. And this adds nucleotides to build that mRNA strand. Just remember that instead of T on this RNA strand, we're going to have U. All of this takes place in the nucleus. So you see here that transcription is taking place. We're taking that DNA code and we're converting it to mRNA. That mRNA is then going to ship outside of the nucleus and head towards the ribosome. So again, the process is very similar to DNA replication. The only difference is that we're making RNA instead of DNA. And that RNA is single-stranded and going to leave the nucleus. Now, a lot of that RNA doesn't really code for anything. It's what we call junk DNA or junk RNA. It's simply just RNA that doesn't really code for anything, so we don't need it. So there's a process inside of the nucleus called splicing. And splicing removes the part of the RNA that doesn't code for anything, that isn't necessary. So what's going to happen is that it's going to be spliced. The intron is going to be removed. Intron is the RNA that is not required to make protein, so it's no longer needed. The exons are left over. The exons are what is required to make the protein. So this process known as splicing removes the intron and pieces the exons together, making RNA that is going to be coding for something important. All right? The intron is not important, so it is removed. The exon is left behind because it's going to code for something. That RNA is going to leave the nucleus and head towards the ribosome. Now, in order to understand the process of translation, we have to understand how that mRNA strand is going to be read. The sequence of DNA codes and RNA codes are for specific amino acids. So you look here, we have a table of RNA codes, and you notice that there are 3 letters for each code. Each 3 nucleotide sequence are called a codon, and each codon codes for 1 amino acid. So when this process of translation occurs, we're going to take that sequence and read off 3 nucleotides at a time. Each of those 3 nucleotides is going to code for a different amino acid. So you notice that there are 20 different amino acids and that several codons overlap. So for example, if we read that mRNA and read CUU, you see that it says LEU or leucine. It's the same thing for CUC, CUA, and CUG. So several of these amino acids are coded by different 3 nucleotide sequences. This will be important to understand when we talk about genetic mutations. But notice that there are 3 stop codons, UAA, UAG, and UGA. This code stops the process of translation. And that's important as well, because if we have a finished protein, we don't need to continue reading that sequence. Additionally, you see that AUG is the only one that codes for methionine, and this is the start codon. Once this is read, the process of translation will start. So just a couple of key things we need to know about those codes and the sequences that make those amino acids to build those proteins. Again, I understand it's a long process, but it's important to understand. So the process of translation. The mRNA, again, is going to leave the nucleus and head to the ribosome. Once at the ribosome, the tRNA is going to attach amino acids together to form a protein from each codon on the mRNA. tRNAs exist in 3 nucleotide sequences, just like the codons do on the mRNA. The tRNA has anticodons that are going to match up to the codons on the mRNA. The start codon, AUG, starts the translation, and the stop codons, which we've talked about, stop translation. So once AUG is read, the tRNA will attach to it and leave behind an amino acid. And each 3 nucleotide sequence will leave behind a different amino acid. These different amino acid sequences form different proteins, and these proteins are the proteins that control most, if not all, of the bodily functions and the characteristics that exist within the body and cell. So this process is extremely important and complex. We'll talk a little bit more about it later, but understand that those tRNA anticodons match up to the codons on the mRNA. Each time it matches up, it leaves behind an amino acid. Those amino acids are bound together through what are called peptide bonds that create a protein. And that protein is what we want to look at. So So just real briefly to analyze the code, well, if we have a DNA sequence that is TAC, GCC, AGG, ATT, the complementary mRNA sequence is going to read AUG, CGG, UCC, UAA, right, using the base pair rules that we've talked about in the previous lecture. A goes with T, C goes with G, and just remember that with mRNA, A will go with U. When we break it down into codons, remember we break them down into 3 nucleotide sequences. So we have AUG, CGG, UCC, and UAA. We then look up the amino acid sequence on the chart to see what those codes code for. AUG codes for methionine, CGG is arginine, UCC is serine, and UAA is the stop codon. So we can see how we take that DNA sequence and make those amino acids from that DNA sequence. It requires a little bit of work and a little bit of thought, but it's pretty straightforward on how that goes. So hopefully you understand a little bit more about the central dogma of biology and the fact that DNA codes for RNA, and that RNA then codes for proteins. You should also be able to describe protein synthesis in 2 steps: the process of transcription, making that mRNA from the DNA, splicing that mRNA to leave behind the exons that we want, and those exons within that mRNA head to the ribosome and get read by the tRNA, which leaves behind an amino acid sequence that is used to build the protein. Hopefully you understand this a little bit better. We'll discuss this in detail a little bit later on. Make sure you take your video quiz, and have a good day, guys. We'll talk to you later.